It says beetroot tissue was cut into pieces of similar size and shape. The masses of the individual pieces were measured. 3 pieces of beetroot were placed in each of 6 sucrose different solution. After 3 hours, the pieces of beetroot were removed from the solution, surface dried, and their masses recorded. The mean percentage change in the mass of the beetroot in each solution was calculated and displayed in the graph. This is a nice question to get, because what is it a lab about? Osmosis, right? We've all done an osmosis lab once in our science career. So it's actually a good one to get because you understand what's going on. So here we can see the change in mass in percentage. And here we see the concentration of sucrose. So depending on how much glucose or the sucrose was in this solution, the change in mass of the beetroot was higher or lower. Okay, and here it tells you the change in mass, if it gained mass or if it decreased mass. We're going to go ahead and do the first question. It says, state the independent variable in the experiment. So just remember independent variable, but I'm going to go up so you can use the data to help you. So independent, this is SL. Independent variable. And once you answer what the independent variable is, can you define what the independent variable is by definition? Someone said the concentration. Okay. Is anyone thinking something else? Concentration of sucrose. Very good. Anyone else? Concentration. Nice. Okay, good. If you said concentration, you did get it right. It is the concentration of sucrose. And what is the definition of independent? One thing that will help you on the exam, which is nice, is that the independent variable is usually on the x-axis. So if you ever have trouble just remembering what an independent variable and dependent variable is, you remember that independent is always going to be on my x-axis, and then dependent is going to be on my y. And someone in the comments did say this is the variable that you're manipulating. Very good. So this is the variable that I'm manipulating, and then the dependent is the thing that I'm measuring. Okay? So for this answer, all you need to say to get the mark, it's 1 mark, you just say sucrose concentration. Okay, and of course, write proper concentration. I just wrote CUNC as short terms. Okay, next it says outline the reason that beetroot pieces of similar size and shape are needed in order to obtain reliable results. And this is a very famous 2 words that IB love using. Don't you need the units? No, you do not need the units to get the mark. Good question. I'm just gonna go to the side and get my charger to reduce error. Okay, but what would cause that error? Is this prediction paper? It's not a prediction paper, it's the specimen paper. So it's written by IB, who kind of give, you're not supposed to get this as a student, it's only supposed to be available to teachers, but it's written by IB to tell the teachers how it's going to go. But everyone knows that the specimen paper is usually much easier than the actual exam. Like you should expect this to be a bit easier than your exam. Thicker size have different effects and longer diffusion. Very good, exactly. So thicker size, you are correct. You can say thicker size. And so the size would affect the diffusion. Perfect. And can you elaborate more? What is this? The size, so the mass will change with size. That can be 1 more. Can anyone name another one? So we know that IB love using surface area. When in doubt, literally talk about surface area. Because IB just love using the idea of surface area. I'm not sure why. It's not really a misunderstood concept and you would think it is. But the IB love talking about surface area. So, of course, different surface areas would affect the rate of diffusion. Okay? So how do you get the mark? To get the mark, you have to name a variable. You can talk about size. You can talk about surface area. But to get the second mark, you have to explain why it's affected. So if you just said size or surface area, you would only get 1 mark. So you must say surface area would vary or change, whatever you'd like to say, osmosis, or someone said diffusion rate. And so 1 mark is stating the variable, and the second mark would be explaining the variable. So that variable needs to be controlled. How does the surface area affect it? Okay, so if you have a larger surface area, okay, that means more of the water molecules can seep out, okay, through the available surface area, or more of the water can seep away, okay, because if this was a very low surface area, it would, like, the water would go out much slower, and that's why it would affect. Very good. Anya said larger surface area increases exchange rate. Does this make sense, everyone? Is this math? No, it's biology. Is this physics? No, it's biology. It's bio-SL. So if you're a bio-SL, please stay along. I'm going to move on to the next question. Suggest a reason for expressing the changes in mass as percentage. So what is the reason for expressing the changes in mass as percentage? What could a percentage show us? Thank you guys so much. If you're answering the questions, helping me out in the chat, I really appreciate it. To determine the change in mass, nice. You're right, but you have to phrase it in a more obvious way. IB like when their students are obvious. So you're right, you're talking about change in mass. So you're talking about a change in something. So you're talking about a start and an end. That's what they just want you to say. They want you to say with a percentage, you're able to see the difference from the start to the end. So be very stupid. Imagine you're explaining this to a toddler, because you, as Tay said, that's exactly what you would say. You'd say change in mass, but you have to be very, very obvious, because you would say that there's a beginning and then there's an end. So you can say allows for comparison of initial plus end, or you can say versus end, whatever you'd like. And that's all you would need to get the mark. Concentration between different concentrates, is there a possible mass difference? It would not be to disregard the possible mass difference. To express the difference in two samples. I did already solve the paper 1A of the specimen paper, and it's on YouTube if you want to see it. Does this make sense, everyone, where I'm saying the initial versus the... So you can compare, basically. But if they have... Okay, so someone said if they have different starting masses, they cannot have different starting masses. That's a control variable for this experiment. They should not have different starting masses. Because remember, the only two things you can change, or the only thing you can change, is the sucrose concentration. This will also be uploaded on YouTube. Okay, so comparisons of mass from start percent, exactly perfect. In like simplified terms, literally change of mass, but they want you to be obvious. Could you explain what SA is? SA just means surface area. So the surface area, like volume, versus the surface area of a cube. If you want me to go and explain that more, I can. But while you answer, I'm just going to move on to the next question. It says, explain the effect of 0.0 mole sucrose solution on the water potential of the beetroot pieces. Water potential is part of the new syllabus content. So hopefully you guys understand what water potential is. Water potential just basically refers high osmosis rate as water potential is the highest in pure water. Water moved to area of high concentration. Very good, Pilly. That's a perfect answer. Water enters the beetroot tissue by the osmosis. There's an increase in water potential. Perfect, guys. So exactly as you're saying, water enters the beetroot. So that would be the first mark. The second mark is just say, literally, this happens via osmosis. The third mark would be to just say, because there is a increase in potential. So, increase in potential inside beetroot. Therefore, water goes in. Because we know water follows solute concentration, just like someone said. Okay, so if water follows the solute concentration, that means it's going to go from the outside in. Can you explain the question answer a little bit more? Of course I can. So basically what's asking is that what is the effect of 0.0 mole sucrose solution on the water potential of the beetroot pieces? So it's telling you when the sucrose concentration is 0.000, so around here, what's going to happen to the mass? Sorry, my alarm. What's going to happen to the change in mass? Here we can see that the change in mass increases. So the beetroot increases in size. Its weight increases. Why? It's because the water is entering. And all you have to say is just explain that the water is entering via osmosis. You're going to explain that this is because water potential is low inside, and therefore water will go from a high potential to a low potential. Because we know by definition, when we have solute in the solution, that means we have low water potential. And we always know that things go from high to low concentration. If this is a bit of a difficult concept to understand, try reviewing water potential, because I know it will be hard to do without understanding. Okay? All right, is this BioSL? It is BioSL. Okay, perfect. I'm going to move on to the next one. So we're already on question 2, which is a good sign. It says, in an experiment investigating the secretion of proteins by glands, researchers supplied radioactive labeled amino acids to a sample of pancreas cells that secrete digestive enzymes. The graph shows the relative abundance of radioactivity labeled proteins in 3 different organelles during the period after the cells were exposed to radioactive labeled amino acids. Okay, I'm going to give you a second to digest this information because that was a lot. So what do we have? We have radioactive amino acids to a sample of pancreas cells that secrete digestive enzymes. So here's my pancreas. These pancreas cells secrete digestive enzymes. So they secrete enzymes that break things down. And what did they do? They added radioactive amino acids to these pancreas cells. So what can we assume? We're going to assume that these digestive enzymes released by the pancreas are, of course, going to digest the radioactive enzymes. So hopefully that makes sense as a concept so far. Sorry, I'm just editing the screen because it got a bit small so you guys can see better. And then it's showing us in our picture here how much of the amino acids were administered and then which organelles secreted what from our pancreas cells. So the graph shows the relative abundance of radioactive labels in 3 different organelles during the period after the cells were exposed to the radioactively labeled amino acids. So this is the incubation time after exposure to radioactively labeled amino acid, and here's the location of radioactivity labeled proteins. Okay? Does this make sense? So radioactivity labeled proteins. So these pancreas cells, they digested the radioactive labeled amino acids, and they absorb these amino acids, okay? And we're going to see where they traveled because we can follow and monitor this radioactivity. So by monitoring this radioactivity, we're able to see where exactly amino acids, when they get digested, where they go. Does this make sense, guys? Please let me know in the chat if that makes sense or you want me to re-explain. I have to examine 1 year. I'm cooked. No, you're fine. You have a year. You're good. Don't worry. Let me know if that makes sense. And if it does, I'll continue to the questions. If it doesn't make sense, I don't get it. Okay, let's go ahead and explain it again. Oh, my screen keeps... Okay, good. So, let's start again. So basically, what is it saying? It's saying that a pancreas cell, here's our pancreas. This is my bad pancreas, whatever. My pancreas cell secretes digestive enzymes. And basically what the scientist did, the scientist said, okay, I want to see what happens when we digest proteins, what happens after that fact. Okay, so what did he do? Imagine the pancreas. He said, I'm going to introduce amino acids to this pancreas. Naturally, this pancreas secretes digestive enzymes to break down these amino acids and reabsorbs the amino acid for their benefit. Because we know in the digestive tract, what's the point of the digestive tract? The digestive tract, the point is to digest the stuff and then absorb it for nutrients. The pancreas is just doing exactly that. So the pancreas is going to break it down and take it in. Now, the key thing here is that they're digesting the amino acid, absorbing it, but it's radioactive amino acid. So we can actually monitor the amino acid as it's moving through the pancreas, okay? So if it's moving to the pancreas, I know exactly where it's moving inside the pancreas. So the point is that this pancreas digested the amino acids, took in the amino acid, and then used the amino acid to create proteins. And once it took proteins, created those proteins, we can see where the radioactivity traveled. Okay? So after 117 minutes, this is how much radioactivity of the proteins were there, how much of the secretory vessels had the radioactive labeled proteins, and so on and so on. Does this make sense, everyone? Does that make a lot more sense or no? Can we explain it again? Don't worry. Can you promise me that you're going to do the same thing next year? So we are going to do the same thing. It's probably not going to be specimen paper. It's going to be the M25. It makes sense? Okay, good. Someone said, is this HL or SL? It's SL. Please share the live if you guys can. We appreciate all the... Oh, and someone sent me a heart. Thank you guys so much. And let's set a like goal too. Should we do 10,000? Yesterday we did 10,000 and we actually got to 20,000. This is biology. Of course, I'm happy to do this. Okay, let's have a look at the question. It says, suggest a reason for using amino acids that were radioactively labeled. So what's the point of doing radioactively labeled? I kind of gave the answer in my explanation. But what's nice about the radioactive proteins? We can see what... How many questions do you do in the crash course? I don't know, hundreds? I don't know how much we do in the crash course. A lot. To track their movement. Perfect. Wow, that was a really good answer. It's easier to track, keep control of them. Very good. To see where it ends up. Perfect, guys. As long as you mention track, you're good. To see where they end up, where they go, you're able to see the movement. Exactly. Very good. And that's all you need to say to get the first mark. So of course you're going to put it in sentence format because it's just route to right track. But you all got the answer correct. Next it says, X is a membrane-bound organelle. Identify X. So remember, we are in the pancreas, and we're talking about something that deals with proteins, that is not a secretory vessel, that is not the Golgi apparatus. So what can we assume it is? It's part of this group. Someone put the right answer. I'm not going to say who. So someone said Vest, careful, it's not. It has to be a specific membrane bound. Now, someone said ribosome. Be careful. Is a ribosome always, is it membrane bound? And is it always in this structure? Or does it only assemble when you try to make proteins? So we cannot say ribosome because it's not always a ribosome. Very good, guys. Most of you said rough ER. Exactly. Why? Because we know rough ER is membrane bound. And we know that it's part of this movement of proteins. What are the organelles that are, part of the proteins? We have the Golgi apparatus, we have the rough ER, and we have secretory vessels that then secrete all the things you're making from the rough ER. Does this make sense why the answer is rough ER? But very good, guys. Most of you got it. We're almost at 5K likes. Thank you guys so much. Good luck studying for your German. Okay. Yes, okay, good. What do we track? We track the proteins. So it's showing you the location of the radioactively labeled proteins. What are your best tips for studying for IB biology? Definitely find a way to cram the content. So I always recommend the crash course, but if you don't want to buy the crash course, you can always just watch YouTube videos to cram the content. And then once you cram the content, do as many practice questions as you can. But for bio, definitely recommend taking time to learn the content fully, because it's really about how well you know the content. But for chemistry, for example, I would just recommend going straight on practice questions. Okay, next it says, explain the role of ribosomes located on X. So we've identified X as rough ER. What is the point of the ribosomes on the rough ER? Nice. Someone said, I don't want to say the answer and giving you guys, I didn't understand the tracking thing. So basically, create a protein, make a protein synthesis. Very good, guys. Exactly. That's all you need to say. Let's see how many marks. If it's 1 mark, that's all you need to say. Keep an eye on the marks, because if you know if it's usually 2 marks, you have to say something and explain it. Okay? Unless they, for example, say state 2 things, then of course I have to state 2 things. But if it's basically them saying state 1 thing, and it's 2 marks, you have to not only state it, but you have to explain it. What subjects is this? This is bio. Okay. Next, it says gland cells that secrete large volumes of fluid typically have many aquaporins. Hopefully, you guys are familiar with this in their plasma membrane. Suggest a reason for this. Now, if you haven't heard of this, I want you to use your knowledge of languages. What does aqua mean? This is IB, not GCSEs. Wait, someone said ribosomes carry out photosynthesis. They do not carry out photosynthesis. Careful. Diffusion of water to let water leave. Water. Exactly. We know aqua means water. Exactly. Very good, guys. So you know that an aquaporin is actually a channel protein that allows water to diffuse through. Why? Because we know our phospholipid bilayer, this is a good time for active recall. We know our phospholipid bilayer has What type of heads and what types of tail? What are their characteristic? A hydro what and a hydro what? A hydro. Which one's hydrophilic? Which one's hydrophobic? Tell me in the chat as active recall. Nice. So which one's hydrophobic? The tails are hydrophobic. Exactly. Very good. Hydrophobic. So you can imagine when water wants to move into my plasma membrane, it can't because of those hydrophobic tails. They're non-polar. They're fat, like fatty acids. So we know we need to have a protein channel to help me exactly like move my water inside. Okay? That's why we know aquaporins are for that benefit. So suggest a reason for this. What can you guys say for a reason for this? Water diffuses through the membrane, so that's 1 mark. How many marks is this? Let's see. It's only 1 mark, so you can just say water diffuses the membrane because of this non-polar area. And that's all you need to get the mark. Thank you guys for almost 6K. We're almost at 6K likes. Let's try to get as high as we can. Okay. Does this make sense? I'm going to go ahead and move on. It says, after synthesis, globular proteins assume their final tertiary structure. And this is a bit mean of them to ask on an SL paper because we know this is more of HL content. You go in much more detail in this in HL. Explain the relationship between the sequence of amino acids and the tertiary structure of globular proteins. So this was name dropped in the old syllabus, but it's highlighted as a whole syllabus point in the new syllabus. So this is a topic you should know, or hopefully your teacher should have taught. But if not, and I know some people in this live are doing the crash course, and I remember teaching this topic. Where did you find this past paper? It's not a past paper, it's a specimen paper. If you just search up IB biology specimen paper, it should show up. Very good. Someone said, sorry, I can't pronounce your name, but you exactly said R group interactions determine the tertiary structure. Can you give me a bit more detail? What about the R group? What do you have to think about when you're thinking about globular proteins? And this is 4 marks, so you have to say a lot. And a good IB tip I like mentioning to my students always, when this is 4 marks, I want you to mention 6 stuff. I want you to mention exactly 6 points that you can use to explain this specific question. Because one thing about IB is that they don't remove marks, they just tick off the correct things. So let's say hypothetically you miswrote something because you are tired on the exam day, you're so stressed. That's fine. They're still going to mark the 2 extra ones that you mentioned. Okay? They're going to read no matter how long your answer is until you get full marks. So be really, really specific. Okay, I'm just going to have it. Isn't the tertiary structure like the side chain interactions and H bonding, etc.? So you could very good. You have to have a bit more detail. So I'm not sure how your teachers taught you this, but I'm just going to teach you this, how it's supposed to be taught. So we know that this is our, oh, this is really ugly, wait. This is our polypeptide chain with my amino acids and so on. I know that some of these amino acids are going to have, I'm going to do it in blue, polar stuff. Polar, polar, polar. And we know polar means they're attracted to water. And some of these have hydrophilic. It depends on the R group. It's all dependent on the R group. And what ends up happening is when you place this protein in an aqueous environment, and aqueous is basically most of the environments in our body, when you place it in an aqueous environment, such as the blood, which is mostly made out of water, what ends up happening is that these lipids basically are trying to hide. So this is my protein. These lipids, or the hydrophobic R groups here, are going to try to hide. So they try to all join up in the middle. And what ends up happening is that R groups of the outer ones will be on the outside. And it will form a structure. I wish I had a picture to show you guys better this, but hopefully it makes sense. That basically all the hydrophilic R groups are going to go on the outside, and all the hydrophobic ones are going to go on the inside to try to hide, to try to go away from the aqueous environment. And therefore, it forms globular proteins. And that's exactly what you need to say in the mark. So what do you need to say? The sequence, you could say sequence of amino acids. I'm just going to say AA, amino acids, determines structure. And just like you said, R group interactions, you have to say they can be hydrophilic, and then hydrophobic. And then what you have to specifically say to get the remaining marks is to say the hydrophilic ones will go on the outside, to interact with aqueous environment, and inner hydrophobic will like hide from the aqueous environment. And yeah, that's what you need to say. Hopefully that makes sense, guys. Is this taught in first year bio or second? This should be, it depends on your teacher, honestly, but it should be taught in first year. Which topic is this? This is topic, I only know it in the topics that we have in the crash course, but I can show you at the end. It's with all the monomers and the protein unit. We don't talk about the polypeptide bond. What do you mean polypeptide bond? Someone said B12. Thank you so much. Yay, we're almost at 10K. They say the same thing for transmembrane proteins. Exactly. You can say, but you have to be very specific that when you're talking about transmembrane proteins, some part of our membrane is hydrophilic and some part is hydrophobic. So you just have to be careful to say that the middle of our protein is hydrophobic, and then the parts that are interacting with the heads is hydrophilic. But you say the exact same thing. Very good. Is it important to talk about the interaction, different charges between the R group itself? Yes, if you mean like which one's like hydrophobic, if you mean by charges, you mean like polar and nonpolar. You don't have to specifically say, you can say polar and nonpolar, you would still get the mark, but you don't have to say, you could just say hydrophilic and hydrophobic. Do these kinds of lives regularly happen? They just happen based on availability and demand. And I know a lot of you guys yesterday requested when I went live to go again, so I did go again when I have time. I usually put the live schedule on the TikTok page. So if you're interested in that, maybe you can give us a follow and then always go on the page and it tells you like when a live is scheduled. Okay, I'm going to move on to the next question. We're almost there. I think this might be the last question. Oh no, we have one more. One more and that's it. We have two more. Okay, perfect. You guys have been doing very, very well. It says, Rhizobium, is a genus of bacteria that live in the roots of flowering plants in the family Fabaceae. Plants form nodules surrounding a population of Rhizobium. The bacteria fix nitrogen from the air into nitrogen compounds. This is also new syllabus, especially ammonia, which are made available to the host plant. So if you do the crash course, you know we've taught you already this example. So what's nice about the crash course is that we tell you the examples IB are going to use, because IB tried to be sneaky. But in the syllabus, if you like zoom in, they tell you exactly the examples you're going to use in the exam. And in the crash course, we already talked about this example. So if anyone can remember, or if you've done this in class, hopefully you have, this example, try to reflect about what was said in class. It says the graph shows changes in the diameter of the root nodule for a plant grown in nitrate-enriched environment and a control group in nitrate-free environment. Outline the concept of mutualism with respect to this example. So the first one's quite easy, but if you don't know the example, it's a bit hard. What level is this? This is SL. So someone said, what about amphipathic? And I'm assuming you're talking about the previous question. If something's amphipathic, it's usually a large molecule. It has to have like an end that's polar and an end that's non-polar. Like for example, steroid, it's a very large molecule. So what you usually see is that the polar side will go like towards the aqueous environment and then the non-polar will go to the middle. Very good, Tay. You've exactly mentioned the relationship. Okay, you do not know this example. Okay, who else doesn't know this example? Because if you don't know this example, I can explain it because you do need to know it. Or it will be used as an example. This is SL level. Mutualism is a relationship where both species benefit from each other. In this case, the plant gives. Very good, Ivan. You've explained it most. Please explain. Okay, I can explain it. Of course. No worries, guys. So basically, with this plant, and generally in plants in general, you have plants like this. Oh, that's a really bad plant. Guys, I'm so sorry. This is my plant in my soil. Here. Wow, I love this drawing. Anyway, and have you guys heard of fertilizers? You know, fertilizers help the plant grow. And usually farmers, they put artificial fertilizers. We actually have natural fertilizers. What are these natural fertilizers? These are the bacteria that grow near the roots. So this is my bacteria. And what does this bacteria do? This bacteria, okay, is a decomposer. It eats up dead organic matter, and it takes up the nutrients from this dead organic matter. And actually what it releases is nitrogen. Okay, and this is very important. Why? Because nitrogen is a very helpful fertilizer for plants. So what does this bacteria do? This bacteria provides nitrogen, and you do need to be able to know how it got this nitrogen. Not in this example, but in your syllabus, you need to know how it got this nitrogen. And in return, because this is a mutualistic relationship, the bacteria also has to benefit. It can't just be giving all its nitrogen. In return, the plant gives it some of its products of photosynthesis. Can anyone tell me what the product of photosynthesis is? What is the point of photosynthesis? Kind of writing the answer. This is biology. O2, very good, to produce glucose. Perfect, exactly. Very good, Tay. You're answering very well. Exactly, to produce glucose. So it will provide the bacteria with glucose so then the bacteria can use it as a form of nutrients. Okay, so I give this nitrogen, it gives me back glucose, and that's the whole mutualistic relationship. So what do you have to outline in this example? Someone did explain mutualism is when two of the species benefit. Now, if they're telling you with this example, you will get the mark if you only talk about this example. So what do you have to say? You have to say the plant gives glucose and bacteria nitrogen. And that's all you have to say to get the marks. Can it use the O2? Good question. I don't think it can, because the whole point is that the plant gives the glucose directly to it, and the O2 is like diffused in the air. So it has to be like directly given to the bacteria. All right. Guys, we hit 11K likes. Thank you so much. Should we try to beat yesterday's? Yesterday we got 20K. Okay, hopefully that makes sense, guys. I'm just trying to have a read to make sure I didn't miss anything because I don't want to. Okay. I'm going to move on. Is this microbiology? It's not microbiology. It's more about the species unit and understanding like the biomes, the ecosystem is that one. Hi, Mati. Okay, we're going to move on to the next one. It says distinguish between the cells in Rhizobium and Fabaceae. So this one is just a knowledge question. So this is the plant. Wait, let me double check. I forgot. No, sorry, this is the bacteria, but they tell you. And this is the plant. So hopefully this is easy. What's the difference between a plant cell and a bacterial cell? One of them is prokaryotic, and one of them is what? Nice, you guys are giving very good examples. So this one, the bacteria is prokaryotic. This one's eukaryotic. Okay, be careful with the command terms. So you should be able to find some sort of command term. If you don't, we also have one given in our crash course if you guys like to join that. But if you don't, you can just search up maybe IB command terms, and you should be able to understand what these things mean. So if this said compare, you have to be very careful. You have to compare. Distinguish just basically means list, so you're good. This one is prokaryotic. This one's eukaryotic. This one has ATS ribosomes. This has 7TS. This one has naked DNA. This one has protein-bound DNA. This one has a nucleus. This one has no nucleus. But you guys have answered this very well. Mitochondria, nucleus, so membrane-bound organelles. Exactly. Very good, guys. We're at 13K likes. Thank you guys so much. And thank you to whoever sent the heart. What is meant by naked and protein-bound DNA? Good question. So naked DNA basically means that the DNA is not wrapped around anything. So you should know that DNA wraps in the formation of a chromosome, like this, right? And the way it's able to do that is thanks to proteins that it wraps tightly around. Does that make sense? Okay, so that's all you need. How many marks is this? This is 2 marks, so you don't even need to say much. But make sure you mention a good chunk for both of them. Suggest a reason that the mutualistic relationship often involves species from distantly related groups of organisms. So why is it important in mutualistic relationships that they don't or that they originate from distantly related organisms. Why is that important? If they were close in relationship of organisms, what would be an issue? What is that type of relationship? Exactly, someone very good, guys. You said histone and H1 histones for stabilization. They don't compete. Perfect. Exactly. Good job, guys. So if they were not distantly related, if they were closely related, they would eat the same things. They would require the same mating grounds. They would live in the same environment. Okay, so we would know that they would compete a lot with each other. Interspecific relationships, very good. Exactly. So they will not benefit from each other. So they have to be very different origins so that there's no form of competition. Okay. So no competition. And there's actually one more thing in the mark scheme that I think is a really interesting one that they bring up. And in this one, it basically says that if they are distantly related, it means they can have a wide range of characteristics and therefore are able to provide each other things that the other one could definitely not provide, if that makes sense. So because they're so distantly originated, they have characteristics that are so different that they are able to provide each other with unique properties. Thank you guys so much for helping answer any questions in the chat, but you only need to say one, and one is enough. Okay, analyze the results of the experiment. So now this is the hardest question. A mutualistic relationship is when both species benefit. Is anyone here DP2? I think most of the students are DP2. Okay, so let's analyze the data. So in this, the data is really, really hard to understand. We did this question in the crash course, and a lot of students were confused. And they were messaging in the chat like, I don't get this, so we had to explain it a couple of times. So take a second to really understand the data because it's actually hard. I'm going to tell you this is a really hard one. But we're analyzing their experiment. And remember what we talked about the relationship between those specific things. Now, analyze this data and tell me what this data shows. So we have the mean root nodule diameter. So remember how we said the nitrogen helps grow the plant. So of course the roots are going to grow. Think of that relationship. Then here it says time and day. Here it says this is the nitrate-rich environment, and this is the control. I'm going to give you a couple of seconds to think about this, and then I'm going to answer it with you, because it is a hard one. Is this HL? No, it's SL. We're at 14K likes, guys. Thank you so much. All right, take your time. This is a harder one. Don't worry. Okay, so having additional nitrate will not change the nodule size. Good. Very good, exactly. Is this also relevant for HL? Yes, it's very relevant for HL. Usually, like the last two questions or the last question is the first question for HL. Exactly, very good. Someone said as days go by, the mean root increases for the one, whereas the other one remains stable. Perfect. You would have gotten the mark if you were just more specific. So don't say the other one. I know what you mean. This is a TikTok live. You don't have to be so specific. Okay. Actually, you guys answered this very well. But yeah, you need to say three things. That's where it gets a bit annoying. Can you guys think of anything else? If not, I can just go ahead and run through the specific answers. You guys were able to say one mark, which is good. So the point of this is that we have a plant, okay? And usually, naturally, what does this plant have? This plant has a mutualistic relationship with this bacteria. And when the bacteria is able to provide it with nitrogen, this plant is able to provide the bacteria with glucose. And that's their mutualistic relationship. Now, when scientists intervene and they add extra nitrogen in the environment, we see that that messes up completely our this mutualistic relationship, because we can see when it was nitrate enriched, when we intervened and placed extra nitrate, the plant did not grow at all, which is not good. So we see that they have a mutualistic relationship, And the bacteria knows exactly how much nitrogen to give the plant, and the plant knows exactly how much glucose to give the bacteria. But when we intervene and add too much nitrogen, we can say that the nitrogen, extra nitrogen, inhibits the growth of the plant. Does that make sense? Please let me know in the chat if that makes sense. What do you mean by root? I just mean the root of the plant. Yes, yes. Okay, very good. So you'd have to mention something along the line of this inhibits this. So if something stays constant, a really good word that IB loves using in the mark scheme is inhibition. Okay, or enhances or promotes, inhibits. Use those words to try to express relationships. And one key advice I would give you is that when IB tells you to state a relationship, don't just say one increases and the other one decreases. Say that this has a positive correlation. This has a positive relationship. This has an inversely proportionate relationship. Use those terms because sometimes the mark scheme are very specific. Okay, so whenever you see relationship, don't just say one increases, one decreases, whatever, and so on. Say positive correlation, negative correlation, and so on. Okay, good. Hopefully that makes sense. So, just like you said, you would mention one mark about how there's a difference between this one, like when you add no nitrogen, it's increasing. When you add extra nitrogen, constant. And then the third mark could be why that happens is because one inhibits the other. And what's a fourth mark? You can talk about how, therefore, you can conclude, because you're analyzing the results, you're trying to come up with a conclusion, that the conclusion is that the environment affects the mutualistic relationship. So they're mutualistic to a certain extent, and the environment can have negative impacts on the mutualistic relationships, and that can give us like insight on how species can decline. You guys do ESS review. We currently don't, we don't have ESS crash courses, but for next year, planning on definitely expanding to more crash courses. So if you're DP1, please sign up to our waiting list for next year's crash courses, and hopefully we'll have ESS by now. Is this Saturday the last day for bio HL crash courses? Very good question. It is, but you can still join. Like every single recording is up there, so you can watch it just like a checklist, like topic 1, and you watch the recording of topic 1, topic 2, and you watch the recording of topic 2, and then you like do the practice questions after. Can you repeat the last point? Yes. So because it's telling us to analyze the results of the experiment, you basically have to come up with kind of a conclusion. So what we're trying to conclude based on this experiment, is basically that environment impacts negatively mutualistic relationships. So because the bacteria benefits from the nitrogen and because the plant benefits from the nitrogen that it gets from the bacteria, and the plant gives glucose that benefits the bacteria, scientists were trying to investigate how far this relationship can go. And they found that when you intervene, that it actually affects this mutualistic relationship. So we know that these specific species are sensitive. Can you do chemical signaling? I can, but our other tutor teaches it better. And yeah, if you really want chemical signaling and you're struggling, I would suggest to buy the crash course because she's like an expert on this one. Okay, hopefully that makes sense. Okay, perfect. Next it says, before the structure of DNA was... Oh, someone already gave the answer. Good job, guys. Before the structure of DNA was known, researchers analyzed the ratios of the DNA bases found in cells of different species. The tetranucleotide hypothesis, formulated in 1910, proposed that DNA was made up of equal amounts of adenine, guanine, cytosine, and thymine. Okay, this hypothesis was revised following the publication of Chargaff's paper in 1951, from which the data is shown in the table. State one example of a purine. So what is a purine and what is a pyrimidine? Very good. Someone said adenine or guanine is a purine. That was a really bad handwriting, but that's guanine. One or two rings. Exactly. Good, guys. Pyrimidine, cytosine, and thymine, and they're smaller than pyrimidines. Pyrimidine is short. Good. Very good. Someone said purine is 2 rings, and then pyrimidine is 1. Exactly. Very good, guys. I like your mnemonics, the ways you're remembering the little answers, which is perfect. Okay. Explain how these results falsify the tetranucleotide hypothesis. So the tetranucleotide hypothesis is basically believing that for every adenine, you have the same amount of thymine, you have the same amount of guanine, you have the same amount of thymine. But here, how do these results change this knowledge? So let's first apply our knowledge that adenine binds with what? Thymine, right? The way I like to remember is that apples in the tree and cars in the garage. So adenine binds with thymine, and cytosine bonds with guanine. Okay. So looking at the ratios will help us, right? There aren't outliers, meaning there are outliers, meaning there aren't equal bases. Exactly. You can clearly tell that the ratios are not the same. So the only time the ratios are 1-to-1 is when we're looking at what? When we're looking at the relationship between AT and GC, right? That's the only time the ratios are the same. So for example, oops. Do we see that? I'm not uploading the paper somewhere. It's on our Crash Course Hub if you want to join our Crash Course, or you just can watch the answers again on YouTube. Do you have to know the hypothesis, or is it given in the question? It's given in the question, but you should know it from class. All right. So basically in the answer, you can just say the only ones that have the same ratio is A and T, okay, and C and G. And that's how you get this one. And you can say the ratios, or like this is mark and a mark, or you can say the ratios of A and G and T and C are not correct, or not the same. And that's how you know the hypothesis is incorrect. Using the data, evaluate the hypothesis that the ratio of purines to pyrimidines in DNA in all organisms is 1. Is that true? Do we see that in all organisms that the ratio is 1? Oh, sorry, I circled the wrong one. Or is it only in some? Really, it was on your bio mock exam. How did your mock exam go? Guys, we're at 16K likes. We're almost 4K away. We're 4K away from 20, which was yesterday's goal. So we can see from the data that it's not, right? The overall ratio pyrimidines and purine is not 1, only in some. Exactly, very good. No, perfect, guys. Perfect. I really like the person who said the chicken proves that all the organisms do not have, because you've given an example, which is exactly perfect. You're already thinking in that database mindset. So the answer would be no. Okay, perfect. Discuss how these results are consistent with the existence of the last universal common ancestor, Luca. This is part of the new syllabus, and our crash course tutor just taught this to the HL students, and she was really annoyed because the syllabus added so much Luca content that was not needed whatsoever, and it was really annoying. How can I join the crash course? You just join via our website. You got a 7 for paper 1A. Very good. Yeah, just do a bunch of practice questions. If you do paper 2 of the old syllabus, it has a lot of database questions like this, and actually I think they're much harder. So try to do as much paper 2 questions to help improve from the past papers. So someone said, what's the difference between purine and pyrimidine? So they're both bases, parts of the nucleotide that help, like they're DNA bases or RNA bases. And one is made out of 1 ring and the other is made out of 2 rings. So the purine is made out of 2 rings and the pyrimidine is made out of 1 ring. Okay, perfect. So can anyone tell me, and you have to mention 2 things of how this is consistent with the existence of Luca. And I see that there's no comments in the chat. I know everyone hates Luca. We're at 16K likes, guys. Hopefully we can get to 20 by the end of this. Wait, can you scroll up? Of course. How high do you want me to go? Like this? Someone said universal code. Good. Perfect. Oh, Tay sent me a rose. Thank you so much, Tay. You made good job. Exactly. Did it provide evidence that we share a common ancestor? You are right. Luca is HL content, but they need you to know what Luca is. This is an SL paper. We all have a TCG. Very good, exactly, guys. This is HL content. But you need to know what Luca is. But they're basically trying to tell you, how can we have a common ancestor and how come DNA is similar in all the organisms? You try to make that connection. Comparable across all organisms. So you see that they're very rude. So exactly. Genetic DNA equal universal. Guys, I really hope you're not getting mad at my handwriting. It's horrible, I know. It's been a long day. Genetic DNA is universal. And then how do we know it's universal? Because bases are the same. Same code, exactly. Very good, Hero. Now, for HL, you would need to know this a lot more. Next, it says, discuss the evidence of the relationship between the genome size and the complexity of the organism. This is a really good one because IB, this is such a common question. And in the crash course, we really talked about how much this is set up as a trap for students. So if you weren't paying attention before, lock in now because this is so common. And the fact that they're still bringing this up in the new syllabus is crazy. Genome sizes happen fast. Very good, Tay. Wow, Tay's locked in. Guys, get up Tay's level. You can general learn. Very good. Kavaya, hopefully I said that correctly. I think you were also here yesterday. We have a test for physics. Oh, hell no. Genome size is not very good, Yusuf. Exactly. Perfect. Exactly. So a lot of IB, I don't know why they think it's such a hard thing to bring up, but they talk about this a lot. That they think that genome size, a lot of people just assume that genome size will equal complexity because we assume like big equals more complex. But you always have to remember the genome size does not equal complexity. I'm just going to copy exactly someone's comment. I think someone put this exact comment, which is cute. Okay, so genome size does not equal complexity. And then someone mentioned something about prokaryotic and eukaryotic. And exactly that would be 1 mark. So eukaryotic equal larger. Can anyone tell me 4 more marks? I thought this was A-level stuff. No, this is IB. How long did you revise for bio for your externals? For externals, is that exams? Sorry, I don't know what externals is. What subject of this is bio? Eukaryotes have introns, very good. And what is the actual... Perfect. I don't know. This was Kawai again. Good job, guys. Eukaryotic equals introns. Perfect. Not all genes are turned off. Perfect. Try to give me 2 more things because remember I told you when the question is 4 marks, try to mention what's introns. Ooh, good question. You guys should know what introns are. So basically your DNA or your mRNA that's copied from your DNA. We know we have DNA. And from your DNA, we have an mRNA copy. Okay? This mRNA copy, I'm going to draw it in green. What actually ends up happening is this in your mRNA copy is modifications before it goes to the ribosome for translation. Okay? So before it goes to the ribosome for translation, we actually modify, we have what they're called as post-transcription modifications that change the mRNA before they go out. What are these changes? You have something called introns and exons. So remember now you're in your nucleus. This is your nucleus. After you've copied your DNA, here is your strand of mRNA. And I want you to imagine the green stuff as introns, the dark green stuff, and the light green as exons. And basically what ends up happening is that you have an enzyme that comes here and it cuts off all of the introns, okay, and removes them from your mRNA molecule. So all you're left with is just exons. And these exons, which is the remaining part, the part that are not introns, mRNA splicing, very good. Someone name dropped that, perfect. The remaining parts that are just exons, EX, stands for exit the nucleus. And once they exit the nucleus, you can think that they go to the ribosomes and they actually become proteins because they code for proteins. Now, this is a common IB question and it actually showed up a lot in the old syllabus. And I think there was one specimen question about it. So make sure you know this very well. Okay. And the way IB would ask is they would show you an image and be like, which part is cut off? And you could label the one only with exons. And you can also think about they can ask you like, sorry, ran out of my brain. They can ask you, which part of the mRNA molecules actually code for proteins. And then they would give you a bunch of options and you would have to say exons. It would not be introns, it would only be exons. Hopefully that made sense, guys. So introns stay in the nucleus, very good. Introns for in, and exons exit. Hopefully that helps you remember. I can't say that. Compartmentalization allows for a more specific function. Very good. That's another thing you can say. Luca is the last common ancestor. Some bacteria adapt to hostile conditions that humans can, even though we have big genome. Exactly, very good. So bacteria are able to adapt and be, you can say, more complex than humans, even though their genome is much smaller. So complexity does not result, and that's a really good example. And if you name that example, you're actually going to mark. Okay. Very good, guys. I'm very happy with the way you guys have answered this. You got most of the questions. So if you're feeling uncomfortable with paper 1B, you actually did very well. And we're actually